Welcome to part one of this two-part video series on autoimmune disease and the microbiome. This is a lot to tackle, hence the two videos. There are a number of autoimmune diseases, and they're becoming more prevalent in so-called advanced and advancing economies. Is that a coincidence? No, it's not. You may think, what does rheumatoid arthritis, Hashimoto's disease, type 1 diabetes, and multiple sclerosis have in common? Well, as it turns out, they have a lot in common. We're going to spend most of the first video covering three main topics which come up over and over again in just about every scientific paper you'll read on autoimmune disease. Topic one will be immune dysregulation. We'll then move on to something called molecular mimicry, and the last of the big three determinants of autoimmune disease will be review of genetic propensity. We'll conclude by visiting what is probably the #1 bacterial culprit for autoimmune disease across the board, the genus Streptococcus. But first, let's start with a little background information. We start with this interesting and comprehensive study out of Sweden. You can see the study design here. I know it's confusing, so I'll summarize. These researchers investigated genetic, environmental, and microbiome factors in a set of 17,055 babies and followed them for many years. Of this large group, 111 went on to be diagnosed with what is called juvenile idiopathic arthritis, JIA, the first of many autoimmune diseases I will reference in these videos. When we look at breastfeeding, we see significant differences between those who developed autoimmune disease versus those who did not. This is one part of the immune dysregulation component, as is antibiotic use, which I touch on all of the time. And here, we can see those two graphed with C-section. And all are categorized by these funny codes here, which is the genetic propensity component we'll get into later. As you can see, depending on these factors, the risk for JIA varies considerably. But I just want you to walk away with the understanding of the underlying themes. Here's another theme I harp on all of the time, PPI use. This is no small study. These researchers analyzed the data for 297,099 PPI patients as compared with non-PPI controls. Look at the dramatic differences in risk for developing a wide array of autoimmune diseases in those who took PPIs. What do PPIs do? They dramatically alter the microbiome for the worse. I have a whole video dedicated to this one topic. I suggest you watch it. Whether it's antibiotic use, C-section delivery, or a lack of breastfeeding, all topics I have videos on, or PPIs shown here. We're setting the stage for immune dysregulation by altering the microbiome. And for our last intro slide, I thought it necessary to present this, as the data clearly shows that females in general have a higher prevalence of autoimmune diseases after adolescence. These researchers had shown in a previous study that plasma levels of soluble CD14 was higher in women relative to men. That's the part of the immune system which helps to dispose of LPS. LPS is that inflammation component of gram-negative bacteria I always talk about. So, the implication here is that women have more intestinal permeability than men do. So, these researchers took things a step further and looked at the cells that line the gut of both men and women. What did they find? They found that women had significantly lower zonular occludens, ZO, levels as compared to males. I've mentioned zonular occludens before. It's a key component which holds the enterocytes together to avoid intestinal permeability. They also showed that estrogen inhibited ZO protein expression in these human primary gut cells. This is where the rubber meets the road, because if you have an autoimmune disease which affects your nervous system, your knee, or your thyroid, how do you think the trigger got there? It's by intestinal permeability. What controls intestinal permeability? The health of your microbiome. With that being said, it is my contention that the most important of these three components is the immune dysregulation part. And an enormous player in this is the health of your microbiome. You'll see plenty of evidence for this as we progress. But as a quick summary, why do I think this? Well, if we look at the other two, it is the only logical conclusion. For example, let's consider the genetic component. And I'm not doubting the genetic connection between any disease and family members. I've helped way too many people over the years to be this stupid. There is a clear connection many times. However, if you look across the board at all of the data, what's called epidemiological data, this connection is not the strongest predictor of autoimmune disease. We'll touch on this later, but just consider this for now. As a species, our genes are not changing, and yet if we look at the epidemiological data across all nations, those nations which are considered advanced or which are advancing see skyrocketing rates of many so-called Western diseases. Genes do not explain this. The third key component to autoimmune disease is molecular mimicry. This is where the immune system becomes confused and attacks its own tissue. The best example I can think of is the proven connection between multiple sclerosis and Epstein-Barr virus. Multiple sclerosis is a chronic demyelinating autoimmune disease of the central nervous system. EBV, Epstein-Barr virus, establishes a lifelong infection in more than 90% of adults worldwide. Approximately 2.8 million people have MS worldwide. But wait, there are over 8 billion people on the planet. 90% of 8 billion is a whole lot more than 2.8 million. So clearly, molecular mimicry cannot be the major component of autoimmune disease either. Otherwise, the prevalence of MS would be infinitely higher. No, it's immune dysregulation. So let's talk about that. As I've said before, I find the immune system to be mind-numbingly complex. The intricacy and crosstalk, the breadth of molecules, and the long series of events they go through to affect an outcome make me feel stupid. Hundreds of millions of years have created this system which we think we can keep outsmarting with drugs. But instead, we should just help it do its job. With that said, we're going to focus on a tiny but important relationship in this vast sea of complexity. When it comes to autoimmunity, it is well recognized that there is an imbalance between two types of T cells. We're going to focus on those two here. If these two are in balance, you are more tolerant to a perceived threat and insult. However, when these two are out of balance, then you become hyperreactive. Both of these immune cells share a common source and signaling pathway, at least to start. However, in a pro-inflammatory state, certain signals, and I always talk about signals, from pro-inflammatory cytokines to include TNF-alpha, which I cover on occasion, drive Th17 cell proliferation. Th17 cells, in turn, produce a host of inflammatory interleukins, such as interleukin 17, which further drives the inflammation process. This is great when you have an acute infection to fight off. However, when you have a chronic smoldering fire, then this is not good. What is that fire? How about dysbiosis? As these authors state, quote, pathobionts try to attach to or invade intestinal epithelial cells, thereby triggering innate signaling pathways, which then tip the Th17 Treg balance towards Th17 cells. But if we're not fighting off an acute infection, then we want a more laid-back immune system, a more tolerant one. We want fewer of the Th17 cells and more of the Treg cells, or regulatory T cells. These guys drive tolerance. So if inflammatory signals drive the production of Th17 dominance, then the opposite of inflammation must drive regulatory T cell dominance, right? Right. Part of this equation is not having bad bacteria driving inflammation. But how do we do that? We do that by having a healthy microbiome which keeps the bad actors in check. Not only do the good bacteria prevent the bad from driving this cycle of inflammation, but they also produce short-chain fatty acids, the key one being butyrate. Not only does butyrate do many amazing things, see all of my videos, but in the context of autoimmune disease, it promotes the Treg dominant environment and recruits Tregs to the gut. These Tregs produce the anti-inflammatory interleukin-10, something I also mention on occasion. This butyrate-driven Treg development can come from the thymus or can come from outside of the thymus, both of which seem to be important in immune system tolerance. And which bacteria are driving this health-promoting cycle? How about these guys here? As these authors state, Clostridium species belonging to clusters 14A and 4 are also strong inducers of the accumulation of colonic Treg cells. But what are these two clusters? Well, thankfully, you have a microbiome expert to explain to you that these old terms are still in use. But if you follow my videos, you already know these players, just by different names. Welcome to Clostridial Cluster 4, aka Rheumatococcus / Oscillospiraceae, according to the new reclassification, and to Clostridial Cluster 14A, aka Lachnospiraceae. These are both families which house most of the great universal or almost universal health-promoting taxa of the gut. These health-promoting bacteria have been found to be consistently, significantly higher in healthier cohorts and significantly lower in the unhealthy ones across all diseases. The darker the green, the stronger the data as a health promoter. Out of 1,000 or more species in your gut, this handful plays an enormous role in your health. You've probably seen this slide several times before in my videos. Via repetition, there are very familiar names in here, names such as Rheumatococcus, F. prausnitzii, Roseburia, E. rectale, Carpococcus, and others. It is these health-promoting, butyrate-producing, oxygen and antibiotic-sensitive bacteria which are the ones you should be focused on taking good care of. And if you do, they will, in all likelihood, take good care of you. And in the context of autoimmune disease, these players drive authors to make statements like these. You can pause the video here to read these on your own. So to see this in action, I just have one study with the intense title of Faegalibacterium prausnitzii skews human DC to prime IL-10 producing T cells through TRL2, 6 junk signaling, and IL-10, IL-27, CD39, and IDO1 induction. Just to give you an idea of how complex this immune system research can really get. But we're going to simplify this and stick with things that we already know. F. prausnitzii, TNF-alpha, and interleukin-10. Here in figure 2, I've highlighted their text, which talks about how DC, that's dendritic cell, another immune system component, when exposed to F. prausnitzii, produces immunoregulatory molecules. And highlighted in A, we see a huge jump in our anti-inflammatory friend interleukin-10. And in figure 4, I highlighted how F. prausnitzii switched the profile from pro-inflammatory to anti-inflammatory. In B, we see TNF-alpha, that inflammatory cytokine targeted by so many drugs. There is a huge difference in dendritic cell TNF-alpha production when exposed to LPS as compared to LPS plus F. prausnitzii. And to wrap up this section, I have a quote, which states, quote, several commensal bacteria, such as Clostridium species, think Clostridial cluster 4 and 14A, dietary compounds, think short-chain fatty acids, and AHR, ligands participate in the maintenance of tolerance by inducing gut Treg cell responses or by imprinting tolerogenic features on dendritic cells. And again, by yet another set of authors, we see an increase in Th17 listed with a few autoimmune diseases, certainly not comprehensive, and a reduction in regulatory T cells, also associated with a couple more. And lastly, as a transition, they have included into their autoimmune list of suspects this thing called epitope, or molecular mimicry. So let's dive into that. Now, when it comes to the topic of molecular mimicry, clearly there are many who know much more than I. But we don't need to get into extreme details for the purposes of this presentation. I just want you to walk away with a general idea of how it works. Just a brief pause here in the presentation. If you could just hit like and subscribe, it would really help this channel out. We start with the lupus paper. In lupus, the rate of diagnosis in women is about 10 times that of men. highlighting our previous slide of the effects of estrogen on gut permeability. And speaking of gut permeability, here these authors cite the connection between gut permeability and bacteria or parts of bacteria entering circulation, something I mention about in every video. And here they talk about our main theme for this video, how dysbiosis leads to a hypervigilant immune system with a breakdown of immune tolerance to your very own cells. Up in Figure 1, they cite increased levels of some of the usual opportunistic pathogens I mentioned, such as Streptococcus, Veillonella, and R. navis, as being problematic in lupus. And below that, the passage of bacterial components such as LPS into the bloodstream, which can confuse the immune system and cause antibody production against your own tissue. But why does the immune system get confused? Well, because their amino acid sequences, called epitopes, on parts of these viruses, bacteria, fungi, and even food peptides, which look identical or almost identical to amino acid sequences that we have in our own tissue. For example, if you look here at this amino acid sequence from a protein on Klebsiella pneumoniae, it looks very similar to this sequence here on one of our human proteins, which has relevance in systemic sclerosis, another autoimmune disease. The research shows that bacteria and viruses are the prime sources of molecular mimicry. As we see in Table 1, these authors highlight a number of connections. For example, across the top row, we have in column 1 the autoimmune diseases. They're listed there, followed by the human protein that is being attacked by the immune system. Then we have the amino acid sequence identified from that protein that looks a lot like an amino acid sequence from the pathogen, which is column 4. Column 5 is the protein from the pathogen where that epitope comes from. Then we have simply whether it's a bacterium, a virus, or a fungi. And lastly is the amino acid sequence, aka epitope, from the pathogen. So let's look at one of the rows. Here we have lupus. The human protein has an acronym of SMD. On that protein, we have this amino acid sequence. The suspect pathogen here is Epstein-Barr virus. And on one of its proteins, it has this amino acid sequence, which looks very similar to the human sequence on the SMD protein. Now, we tend to think of only the bad guys being problematic when it comes to molecular mimicry, and justifiably so. These authors cite that this whole concept of molecular mimicry began with the well-known connection between Streptococcus pyogenes and the autoimmune destruction of human cardiac muscle tissue. If we look at Table 2, it gives us a brief list of pathogens and their associations to various autoimmune diseases. If we stay on the topic of Streptococcus pyogenes, we see for autoimmune myocarditis, the human protein which is attacked by its own immune system is cardiac myosin, which looks a lot like, in fact 80% like, the M protein found in Streptococcus pyogenes. The role of this protein in this bacterium is that of a virulence factor, something I also mention frequently. Let's look at another example. In rheumatoid arthritis, the human protein that is attacked by its own immune system is human alpha amylase. The bacterial protein that the immune system is trying to attack is bacterial amylase. They not only look similar, but they sound similar. Why? Because millions of years of shared evolution and pathways. This cross-reactivity comes from the bacterium Porphyromonas gingivalis, the #1 suspect in gingivitis. I also frequently talk about oral health. This protein has a role in glycolysis for the bacterium. But in Table 3 below, we see regular resident or commensal bacteria also being guilty of molecular mimicry in a number of papers. Some very common members of the microbiome are listed here, to include 2 health promoters. And here are the immune diseases with which they are associated. Although the vast majority of time, the molecular mimicry is associated with a pathogenic virus or bacterium, how is it that normal commensal bacteria or even health promoters can be associated with an autoimmune response? Well, these authors have a couple of very interesting papers. First, not shown here, they looked at viral and human protein overlap and found such a high degree of overlap that they stated, quote, in conflict to the dominant tendency to causally associate viral infections and autoimmune diseases, these findings support the view that molecular mimicry is overemphasized as a critical mechanism during autoimmune disease pathogenesis. End of quote. So they went on to look at human and bacterial overlap, shown here. They go on to state that the overlap is extreme. In fact, the title of their paper is, quote, no human protein is exempt from bacterial motifs, not even one. That's a powerful statement, but believable, since we do share pathways with bacteria. I mentioned the sirtuin pathway. It's a recognition of feast or famine. Well, this came from bacteria. Even our own mitochondria are believed to come from bacteria. So, at minute levels, there will be shared epitopes. So if the whole autoimmune story were all about molecular mimicry, then everyone on the planet would have an autoimmune disease, instead of the 5% who currently do. I don't want you to think I'm discounting molecular mimicry altogether, because that's certainly not the case. There are plenty of examples where the risk profile is much higher in a given autoimmune disease when an immunological response is present to a specific pathogen. Again, I return to multiple sclerosis and Epstein-Barr virus. In probably the best or one of the best studies exemplifying this, a study of more than 10 million U.S. Army personnel were followed up over 20 years, and a 32-fold increased risk of MS diagnosis was shown in individuals who converted to EBV seropositivity compared to those who remained seronegative. But yet again, I state that 90% of us adults have EBV in our bodies. Why is this late infection not driving MS in everyone? With that question, we dive into the third component of autoimmune disease, genetic propensity. When it comes to genes and autoimmune disease, there is one factor that without a doubt stands out from the rest. It's called human leukocyte antigen, HLA, and accounts for 1/2 of the known genetic predispositions. And although more than 200 associations between HLA and disease, both autoimmune and other, have been described, the exact mechanism behind this is not clearly understood. What we do understand, for decades now, that some of these associations are very strong, and that its role in autoimmunity is so robust that assessing HLA status is an essential role in determining donor-recipient immune compatibility in organ transplantation. So what does it do? Well, it presents peptides, that would be amino acid sequences, like the ones we talked about earlier, to other components of the immune system for identification. Identification of what? To determine if these peptides are from our tissue or from potential bad actors. And these HLAs have a peptide binding groove, which has 2 walls and a floor. The T cell receptor must be placed properly in order to interact correctly with HLA. It appears somewhere in this equation I just discussed lie genetic flaws in the presentation and recognition. And there are 2 types of mention, class 1 and class 2. Class 1 genes are expressed by all nucleated cells. They present peptides derived from intracellular proteins to CD8 T cells, but they can also be recognized by natural killer cells. Conversely, class 2 HLA is only expressed by professional antigen-presenting cells that interact with CD4 T cells. And this class presents peptides derived from extracellular proteins. HLA also plays a major role in T cell selection in the thymus, which is a gland critical to proper immune response. Here we see a long list of autoimmune diseases associated with Class 1 and Class 2 HLA genotypes. You may see your autoimmune disease here if you have one. For example, in ankylosing spondylitis, HLA-B27 is the big culprit. Are you ready for this statistic? Over 90% of AS patients are carriers of HLA-B27, while only 7 to 12% of healthy controls are carriers. That's a huge difference, right? What a smoking gun. But let's put that in a different context. Only 5% of the HLA-B27 carriers will develop AS at some point. So that means that to have the genetic predisposition is in no way a guarantee that you will go on to develop ankylosing spondylitis. Even the concordance rate in homozygotic twins, that means when both identical twins have the disease, it is only about 60%. And if we look at rheumatoid arthritis, HLA-DRB1 is the big suspect. But the concordance rate between monozygotic twins is only about 25%. So that means if your genetically identical twin is diagnosed with RA, that you only stand about a 25% risk of being diagnosed with RA as well. By definition, there are other variables at play than just genes. What did your twin experience in their life that you did or did not experience in yours? And to conclude this slide, why do twice as many females as males have MS? That's not an HLA gene. Perhaps that's related to the intestinal permeability slide from earlier, something you'll see a lot more of in part 2. Now that we have covered the 3 components of autoimmune disease, let's take a look at one genus, Streptococcus. This is a large genus, and some would even say, well, there are 2 species of probiotics within Streptococcus. Yeah, there are 2 species marketed and sold as probiotics, but they don't determine your health, just like the other probiotics. The majority of this genus contains bad-acting species. So, why did I choose Streptococcus? Because if you look at this genus across all diseases/conditions I've analyzed over the years as the former director of medical education for a microbiome firm and now with my own platform, the bad data points for this genus spike highest in autoimmune disease. And to go back to a previous slide, this is where the concept of molecular mimicry got its roots. Let's begin our Streptococcus conversation by understanding that there are an enormous number of species within this genus. They are actually grouped according to their carbohydrate composition of their cell walls. Here in Table 1, we see a number of diseases associated with Streptococcus via molecular mimicry. Let's take primary biliary cirrhosis, for example. Several bacterial strains and viruses have been proposed to cause PBC via molecular mimicry. And 2 key ones are normal members of the microbiome, E. coli, a name I always mention, and Streptococcus intermedius. Or, if we consider pediatric autoimmune neuropsychiatric disorders associated with streptococcal infections, PANDAS, we know that it's a sudden onset obsessive-compulsive disorder accompanied by a previous streptococcal infection. Symptoms include separation anxiety, nighttime fears, cognitive deficits, oppositional behaviors, and hyperactivity. It has been hypothesized that via molecular mimicry, M proteins trigger the production of anti-neuronal antibodies, such as antibodies against both dopamine 1 and dopamine 2 receptors. And speaking of M proteins, this brings us back full circle to the well-established condition where molecular mimicry got its start. The easiest way to think about Streptococcus pyogenes is as strep throat, something with which we can all associate. Now, in almost all of us, strep throat resolves and we go about our lives. But in some, acute rheumatic fever develops. This is an autoimmune response to an S. pyogenes infection. It may develop approximately 2 to 4 weeks after the pharyngeal infection and is characterized by inflammation of the joints and the heart, which brings us back to the M protein. When our immune system binds to a protein, antigen, it isn't binding to the entire full-length protein. Instead, it is binding to a segment of that protein known as an epitope. In general, an epitope is approximately 5 or 6 amino acids in length. And this M protein epitope looks a lot like some of our own heart proteins, hence the autoimmunity to the heart, which results in about 275,000 deaths per year. Strep infections can also cause post-streptococcal reactive arthritis attacking connective tissue. The point being that whether strep is a normal member of the microbiome escaping via gut permeability and going systemic, or whether via a community-acquired illness in those who are susceptible, immune tolerance being the main factor, bad outcomes can happen. But the rest of us get along fine with strep species in our guts and with exposure to the occasional illness. And speaking of community-acquired illnesses, we have come full circle with this last slide. Let's take the species Streptococcus pneumoniae. It's the main cause of community-acquired pneumonia, or CAP, on this slide. If you were to get CAP, you have a whole lot of S. pneumoniae in your body for immune system to deal with. But there's another factor. Infections like CAP and COVID drive dramatic changes for the worse in the gut microbiome. Here we can see how the flu, tuberculosis, CAP, and respiratory tract infections negatively impact the gut microbiome, probably in part via the gut-lung axis. We can see consistently these infections have been shown to significantly increase known opportunistic pathogens within the genera Enterococcus, Escherichia, that's E. coli, and Streptococcus. And conversely, they consistently, significantly decrease known health-promoting genera such as Eubacterium, Faecalibacterium, Roseburia, and Rheumatococcus. So now, not only do you have to deal with Streptococcus, but you have immune dysregulation because your microbiome has become dysbiotic. Now the chances of a hypervigilant immune system cross-reacting with your own tissue is increased due to the inflammatory changes we discussed earlier between regulatory T cells and Th17. This is why epidemiological data shows an increase in the incidence of type 1 diabetes, another autoimmune disease, after enterovirus epidemics. In fact, one such study measured enterovirus RNA or viral protein in blood or stool, finding that the presence of infection was 10 times more common in children with type 1 diabetes compared with controls. A whole list of other viruses have also been associated with type 1 diabetes. These similar epitopes from viruses and pancreatic beta cells can lead to their destruction and a dependence on insulin. But again, not everyone who contracts a virus will develop type 1 diabetes. It's only a very small portion of us. Therefore, we now know that genetic propensity plays a role in autoimmune disease. But, as we've seen, it is clearly not determinative. We also know that there is plenty of data linking molecular mimicry to autoimmune disease. But yet again, this does not occur in a vacuum. It is the setting of the stage, the health or lack thereof of the microbiome, which determines if your immune response is going to be efficient and tolerant or hypervigilant. It is this immune dysregulation, which is systemic, but at its core originating from the gut, which is the key determinant of whether one will go on to develop a given autoimmune disease while the other 95% won't. For much more reinforcement on this perspective, watch part 2. Well, I hope you enjoyed that video, and I will say that basically every single day on my YouTube comments and in emails, I get a lot of thanks from people out there who really appreciate the information I've been sharing. And so part of that is you're welcome. The other part is you can contribute by clicking on the super thanks below. And if you're not doing a consultation with me and you're not purchasing any protocols, it's a great way to support this channel. Each presentation, depending on the presentation, but most of them take an incredible amount of time to put together. There's a lot of material. There's a lot of data checking. And so it's just sometimes 50, 60, 70 hours to put together one presentation. And so if you can just click that super thanks, I'd appreciate that, and we'll keep the information coming.